Hello all, welcome back to Dr. Kala Edu. Today we are going to discuss a topic regarding mutations. So what is mutation? We all know that recombinance which is occurred due to crossing over. So the recombinance leads to lot of genetic variation which plays a very important role in evolution. The same way the mutation also helps in the genetic variation which also helps in evolution also, because what's happened in mutation is, what is a mutation is a sudden change, okay, a sudden change in the genetic material. So what is the genetic material? Our genetical material is DNA, inside that where the genes are present. So totally everything inside the chromosome. So on the chromosomes genes are present. So where there is a change, sudden change in the genetical material, it may be a chromosome or inside the genes, which is called as an mutation, right? So this mutation, the term was first introduced by Hugo de Vries. So 1901 Hugo de Vries has introduced the term mutation. Okay, so Hugo de Vries, he is the person who has contributed lot of things in about linkage, crossing over, recombinance, mutation, and this modern biology. So he was more explored everything, all the things what we are learning now regarding linkage, recombinance and these mutations. So in 1901 Hugo de Vries, so he has introduced the term mutation, and he has proposed a theory also regarding this mutation as mutation theory. So he has wrote a book also regarding this mutation theory. Okay, right? So he was working on the plant named Oenothera lamarckia. So this Oenothera lamarckia, which is common name is evening primrose. Okay, so in this only he has done lots of experiments regarding mutations, and he has put forward all the thoughts what he has got from the mutations. So you should remember the plant name Oenothera lamarckia, biological name, right? So here broadly we can tell that mutation can be classified into two types. One is gene mutation and another is chromosomal mutation. So one is gene mutation and another is chromosomal mutations. So these gene mutations, they can be said as also point mutation. So gene mutations also called as point mutation. Why it is called as point mutation means there is a specific change in a gene, a point. Okay, only a single gene means what will happen, only one point is changed, where such mutations we call it as gene mutations. So hence it is also called as point mutation. But when it comes to chromosomal, so in chromosomal mutation, what is the thing is, there will be a sudden change in the chromosomal number or any disturbance in the chromosome. We call it as chromosomal mutations. So number itself may be changed, it may get increased or decreased. Sometimes the chromosome itself is getting disturbed, when we call it as chromosomal mutations. So these mutations can be spontaneous or induced, right? Spontaneous means what? It has taken place naturally, suddenly something has hit and then the changes has happened due to the environmental conditions or some other things, that is called as spontaneous. But induced means what we artificially done is called as an induced mutations. So the things which causes the mutagens are called as the substance which causes the mutations are called as the mutagens. Okay, so some of the agents, physical agents or chemical agents, which are responsible for that mutations is called as an mutagens, and the process is called as an mutagenesis. Okay, so the process where the mutants are exposure towards this called as an mutagenized organism. Okay, so a plant or an organism which is get mutant, it is called as an mutagenized agent, a mutagenized organism. Okay, right. So in this gene mutations. So now we are going to detail about this gene mutations. So gene mutations, again we can tell broadly of two types. So in this, what is thing is a single base, a single point base or a base pair of DNA, or single base or a base pair of DNA, when it is getting a sudden change, we call it as a point mutation. Another thing is in these mutations, something it is going to alter the number of copies. So alter the number of copies. How it may be get altered? That is, it may get altered by some repeated nucleotides. So it is going to get number of copies is going to be get increased. Small repeated nucleotides is another type of genetical mutations here. So here this point mutation, which is we are going to discuss in detail. So a single base pair or a base pair of DNA which is getting suddenly changed, we call it as a point mutation. We are going to discuss here. So what is a base pair? Now you will be having a doubt, what is a base pair? So in the DNA, what is the thing is, we have adenine, guanine, thymine and cytosine. Right. So we have what we have: adenine, guanine, thymine and cytosine. These four nitrogenous bases, these are nitrogen based. Okay. So these nitrogenous based are forming pairs in the DNA. So which is the base pairs. So here what is the thing is, adenine always pairs with thymine. Adenine always pairs with thymine, AT. So guanine always pairs with cytosine. Guanine always pairs with cytosine. But here one more beauty is the thing is, this AG we call it as purine. So this AG we call it as purine and TC we call it as pyrimidine. Okay. So this A and G we call it as purine, T and C we call it as pyrimidine. But while pairing, always A will pair with T and G will pair with C. That is A for adenine, G for guanine, T for thymine and C for cytosine. So you should get remembered as like this box condition. So you will not confused when we are going to read about DNA. So that is, again I will write here A, G, T, C. Okay, so this A always pairs with thymine and G always pairs with cytosine. But when it comes for chemical based, so A and G we call it as purine and T and C we call it as pyrimidine, right? So it's clear now. So now the point mutation. So point mutation, again it is getting divided into two types. One is base pair substitution. So what is substitution? Instead of A, so all these four base pairs only going to get changed. Instead of A, there may be a T or there may be a G or a C, whatever may be. So this is called a substitution, one thing for another. It will be replaced. That is the meaning substitution. So base pair substitution or an base pair insertion. Something is going to get inserted newly. So hence it is called as an base pair insertion. So in this, substitution is mainly next discussed. So in the substitution, depend on the mutations, depend upon the mutations, how it is taken place, we can classify, okay, we can classify it as transition mutation and transverse mutation, okay, transition and then transversion. So what happens in this transition mutation? So in this transition mutation, we call it as a nucleotide substitution. So what happens here? I told you, right? So here what will happen? This purine will change with purine. Okay. So for example, that is A, it is going to change as G. Example I am telling, because what we wrote, adenine always, adenine and guanine we tell it as purine. So here the replacement, a substitution or an insert. Substitution, we are studying about substitution, right? So something is going to get substituted. So here what happens? We are purine to purine. A can be substituted by G. So same way in pyrimidines. Pyrimidine, what will happen? A T may be changed with C, or C may be changed as T. So this happens in transition. Okay, transition, that is in the same type itself it is getting changed. But transversion, so the version changing, we can tell it as, you keep on remember the transversion, so the version itself getting changed. So what will happen? So here the purine will change with an pyrimidine, right? So that what will happen? So this A may be changed with T, or T may change with A. So this happens in this transversion, that is adenine, it may pairs with an thiamine, or else in this pyrimidine it may change with purine. Pyrimidine also it can change towards purine, that is what we can tell, C it may change as G, or G with C. Okay, so this is transition and transversion, I think clear. So next is depending upon this, we have some sort of things are to be listed. So first is, so we, according to this point mutation, what they are telling, so there is a change, a substitution in the base pairs. So how the substitutions can be taken place? Either it may be an addition or deletion. So it may be an addition or deletion of a base pair. We can tell it as an addition and deletion of a base pair, a nucleotide, or a mutation. Whatever words we like, we can use. That is, it may be a addition or deletion of mutation, addition or deletion mutation, or addition and deletion of a particular base pair or a nucleotide, right? So we call, okay, totally we call all these as Indel mutations. Totally we call it as Indel mutations. So this is in and then del. Okay, so in means insertion, and del means it is a deletion. So indel, okay, so that is called as Indel mutations. It is an addition or an deletion of that particular gene or an base pair. Okay, right. So what happens due to this Indel mutations, which we call it as substitution mutation, right? So we call this as substitution mutation. You should remember from the substitution only we are reading this. So Indel mutations, which is also called as substitution mutations. The important thing you should remember here is it affects translation. So what is translation? So the DNA will tell that thing at that time it needs to secrete a protein or it needs to secrete a hormone. So at that time what will happen? The DNA will translate all these things to RNA. So this affects the translation. So copy from a DNA to RNA is called as a translation. So that is getting affected due to these indel or substitution mutations. Okay. So regarding this, we have 4 types of mutations here. So what are these 4 types of mutations? First is synonymous mutation, right? So what is this? First is synonymous mutation. So here what happens is one codon of amino acid is replaced on the same codon of that particular amino acids. As we all know that what is the thing is, gene is arranged in a linear manner on the chromosomes. So what will all will be A, T, G, C. So all these things will be there. A, T, G, C, G, 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 C, 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 like these things, the nucleotides will be arranged in a linear manner. So what will happen is a 3 set is called as a codon. All the 3 sets is called as one codon, means there will be A, T, C, G, 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 U, 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 like that. So this 3 things is 3 nucleotide together is called as an one codon. So here in the synonymous, what will happen is one codon for that particular amino acid expression will be changed into another codon, but it will be on the same amino acid. It will be on the same amino acid. The change takes place in the same amino acids. So hence we call it as an synonymous. That is, synonymous means what? The meaning. We can tell like that, right? So you should remember, synonymous means a change happens in the same amino acids, which is also called as silent mutations, which is also called as silent mutations. So next is non-synonymous. The word itself indicates it is non. Okay, so what happens here? The change will be from one amino acid codon towards to another amino acids codon. Okay, it is inside the same amino acids, but here in the non-synonymous, the change will be in different amino acids. Okay, so the change will be in that different amino acids. Okay, different amino acids, that is one from another codon of another amino acids. This is called as non-synonymous, or it is also called as missense mutations. Okay, so this is silent mutation and this is missense mutation. So another thing is called as an nonsense mutation. What? Something, if you are getting irritated, what we will tell? Nonsense. Like that, it is an nonsense mutation. That is, a particular codon will be acting very good, but stupidly what will happen here is it will change to a termination codon or stop codon. So what will happen? This will have a termination codon or stop codon. So what is this termination or stop codon? So like this, if for a protein expression like this, nucleotides will be there. If the continuously it will be like that, returning, it's going on, the expressions, that is the secretions will be more. So one point it should get stopped. Everything one point it should be get stopped. So naturally in the gene also there are some stop codons or termination codons, that is on that particular end of the nucleotide, which should express for the particular amino acid. There will be a stop codon or termination codon. So till that it will get translated, so that that particular amino acid will be produced, right? So what will happen in this nonsense mutations? So these thing will, the good codon will be changed into termination or stop codon, right? So next is frame shift mutations. So what is this frame shift mutations? Due to the change, due to the shifting of this addition or deletion of these mutations, what will happen in this frame shift means there will be the reading frame of the translation process will be changed, totally changed. If a particular normal protein should be getting expressed, but it is not going to be expressed in such a way, and it will be totally changed. The structure and function, both the, what happened in frame shift mutation, both the structure and function and the functions totally is going to get changed in this frame shift mutation due to the what is the reading frame? What is a reading frame? As I said here, this nucleotides, it's a reading frame. If it is getting suddenly changed, so the translation will be getting affected, and the natural protein which want to be secreted will not get secreted. For example, histone. Histone protein is need to be secreted, means it will not get secreted there. So instead some other different function is going to happen, which is called as an frame shift mutations. So here regarding this mutations, I think everyone will clear this point mutation. So you want to keep on remembering that what is an transition and transversion. So in 1 mark they may ask about this purine and pyrimidine. And another thing is indel mutations. You want to know about what are indel mutations. Same way, what are spontaneous or induced mutations, what are mutagens, what is mutagenesis and mutagenesis. These are the things in 1 mark or 2 mark you are getting expected. This point mutation they may ask to summarize you in 3 marks also. I think you are clear with all these in point mutation. So if you have any doubts, you can comment me in the comment section. I will clear your doubts. Thank you. Thanks for watching the video. Meet you all in the next video.